one of the problems we've had in medicine is the obstacles with routine genetic care. And we've worked with Spartan to develop the world's first point-of-care genetic test in medicine. What you see on the lower panel here is the device that was used for the rapid gene study. It's the size of a toaster, and with a buccal swab, our entire study was actually done by clinical nurses at the bedside with no previous genetic testing techniques. And in a half-hour course, they learn how to use the machine. And with the buccal swab, this is actually put into a cartridge directly into the machine, and within 60 minutes, we're able to identify whether patients actually carry the STAR2 allele and how many copies of the STAR2 alleles they carry if they are actual carriers. The actual goal of the rapid gene study was then to evaluate the first point-of-care genetic test in medicine, and in particular, look at the accuracy of this machine and its ability to provide a tailored therapy in carriers of the STAR2 variant. This is the study design of the rapid gene study. In total, there were 200 patients that were enrolled. They were randomized on a one-on-one basis to either a rapid genetic testing approach versus a standard therapy. And those patients with standard therapy, they continue on clopidogrel at 75 milligrams per day. And then those with rapid genetic testing, as soon as the genetics testing results are available, the STAR2 carriers are switched over to prasugrel at 10 milligrams, and non-carriers are kept on clopidogrel at 75 milligrams. At 1 week, repeat platelet function testing with the P2Y12 Verify Now assay is conducted, and at the same time, we also take a blood sample at this time in order to compare our point-of-care genetic testing versus traditional DNA testing techniques. In the standard therapy arm, we also did a point-of-care testing at the time of follow-up so that we allow a better evaluation of the point-of-care test. This is the primary endpoint of the study. So we were looking at the proportion of STAR2 carriers with an impaired response to antiplatelet therapy as measured by the P2Y12 assay and a PRU cutoff of 234. And what you clearly see on the left-hand panel is that patients receiving the rapid genetic test in combination with a tailored therapy, there were no patients with a PRU of greater than 234. This compared to the panel on the right-hand side, those patients on standard therapy, over 30% continue to have a PRU of greater than 234 after 7 days of standard of care treatment. Of note, when we look at the sensitivity and specificity of our point-of-care test compared to DNA sequencing, the sensitivity of the test was 100%, and the specificity was 99.4%. So in summary, what rapid gene has shown is that point-of-care genetic testing after stenting performed at the bedside by clinical nurses permits an accurate identification of the STAR2 carrier status and facilitates a rapid, tailored approach to antiplatelet therapy. Administration of prasugrel to the identified STAR2 carriers decreased the rate of impaired response. So from a clinical importance point of view, this study is important for several reasons. As all of you know, there are greater than 2 million stent procedures performed worldwide per year. Up to 25% of these patients potentially carry an at-risk gene. And rapid gene is essentially a proof of concept that rapid genetic testing in adherence with tailored therapy is now feasible. And the other reason why this is important is that this would be applicable to all other areas of medicine as well. Thank you.